We are going to talk about another tool of recombinant DNA technology, that is cloning vectors. Now, before we start with cloning vectors, we know one thing, that both plasmids and bacteriophages, which are categorically speaking viruses, they have the ability to replicate within the bacteria without any interference from the chromosomal DNA of bacteria. This is one quality of plasmids and bacteriophages which will be utilized as a cloning vector. Okay, this quality makes them suitable for exploitation as the cloning vectors in obtaining the gene cloning. Okay, now you know what the purpose of cloning vector is. Like you know what a vector is. Vector, we take the example of a mosquito. Mosquito carries the protozoan plasmodium to its host. Okay, it itself is a carrier sort of thing which enables the plasmodium vector, plasmodium parasite to reach to its host. In the same way, we are going to have a vector which would either be a plasmid or a virus, where we take retroviruses as the cases, a retrovirus which will be made silent in its pathogenic ability, and instead it will carry our gene of interest and it would act as a vector. That means this is the place, or this is the agency by which the gene of interest would be taken to the host. In the similar way as the mosquito, being a vector, carries the plasmodium, that is the protozoan, pathogenic protozoan, to the host, that is human beings. Same way the gene of interest would be taken to the host, which would be a bacterial cell. Okay, we talk about host as the next tool, so we'll have a next lesson dedicated to that, but for now we have vectors. Now the vector is plasmid, most suitable vector is plasmid. As we had talked about, the first recombinant DNA molecule that was coined by, or that was artificially made by Stanley Cohen, he had studied in depth about plasmids. So we are going to take the plasmid as the cloning vector. Now we had talked about it being a vector. Now let's talk about the cloning part. Now what is this cloning part? Cloning is the process by which gene of interest is producing its copies. Many copies of the gene of interest have to be produced within the host cell. If there are no more copies, if there are more number of copies, the gene would produce its product in ample amount. So we seek that not only our gene of interest enters the host cell where it should be entering it in such a way that it has the ability to produce its copies. That means it has the ability to clone itself. Okay, what is cloning? Cloning is producing the exact copy. Here we are seeking gene cloning. Okay, out of one of the motives that recombinant DNA technology is seeking, the one important one is gene cloning. Once the gene of interest has been identified, it has been extracted, then it has to be cloned. And where it has to be cloned? It has to be cloned within the vector. Now one property of plasmids is that they can very well clone the desired gene, and they have this ability which is measured in the terms of copy numbers. Now what are copy numbers? How many copies a plasmid can produce if it is put in the host. So the plasmids may have copy number 1, it could range up till 100. So 1 to 100, maximum could be 100 or even more, and we seek the plasmid which has more copy numbers. Okay, greater the copy number, easier it would be to obtain more number of copies, and gene cloning would be easier. And this gene cloning has to take place within the host cell. Now this vector is itself having certain characteristics that it must possess to be a good vector. Okay, what makes a plasmid qualify for being a good vector or a cloning vector? We are going to see those points. The first point that a cloning vector must have, first characteristic that it must have, is ORI. Now what is this ORI? Origin of replication. This origin of replication, if you remember, we have studied in DNA replication as well. It is that specific sequence from where the replication takes place. The enzymes would come, they would identify the particular sequence, and then replication would take place. So the plasmid must have its origin of replication, and it is at this site after which the gene of interest must be introduced. Okay, the gene of interest must be introduced after origin of replication, then only we can achieve its maximum number of copies. Supposedly you introduce the gene to that region where replication does not take place, then the gene of interest will not be cloned. Okay, so we have to make sure that the origin of replication is present, and the gene must be incorporated or introduced after it. Okay, the next point is, after origin of replication, we have the next characteristic, that is of selectable marker. Now, what is this selectable marker? Once the plasmid has been made recombinant plasmid, recombinant DNA plasmid, okay, or recombinant vector, and it has entered the host, this plasmid must be marked by some specific activity that tells us that now the host has the plasmid. Are you getting me? It's not that easy. I'll explain it. See, we know that what happens in this entire process, that ultimately our plasmid, which has an alien DNA inside it, that has to go inside the host. This is what the entire process is. Now we must mark it with some other characteristic or other ability which it originally doesn't have, so that we can make it out that the host has been infected by the vector. Okay, this is what I am trying to tell you, that selectable marker is that characteristic which we deliberately put into the plasmid, that is the vector, we deliberately put it into the vector so that we get to know that transformant has been formed or not. Now, what is a transformant? Transformant is the host cell which has recombinant DNA in it. So, whether the cell is a transformant or a non-transformant can be made out if the vector is given a selectable marker. Now, selectable marker is nothing but an activity that is shown once the vector, that is the plasmid, is given the desired gene. Unless you don't give the desired gene to that vector that would act as a selectable marker, it is not going to show that activity. And once it shows that activity, that means your gene of interest has also gone inside and your selectable marker gene has also gone inside, and from that you can make it out whether the transformant is working or not working. And the last characteristic is cloning sites. Third characteristic that a vector must have is the cloning site. Now, these cloning sites are the sites where you have to put your gene of interest or the selectable marker gene. Okay. Now, for the plasmid to have cloning sites, it means it must have restriction sites, where the restriction endonucleases come and cut the plasmid. Now, supposedly this is the plasmid and this is the gene of interest. Now, a restriction enzyme comes and cuts this in this shape. Okay, these are the sticky ends which are produced. So, certainly it must have a restriction site for that enzyme within itself. The plasmid must also have the restriction site, so that when this gene of interest comes and joins over here, a sticky end has been created. If there is no sticky end being produced, this gene of interest cannot go and join over here. For that, the plasmid or the vector must have the cloning site corresponding to the restriction site of the gene of interest. You must have a site for incorporating something inside it, and that site is being provided by this cloning site, and that has to be common for both the cases. The restriction enzyme could come and work on both of them, and same sticky ends would be produced. So this is all you have to keep in mind about the cloning vector. We have to talk about a specific plasmid, that is PBR322, which is a cloning vector. When we understand about that, then you will get a better idea how this cloning vector works. But for now, you have to keep in mind these 3 characteristics which a cloning vector has, and they must be present in the cloning vector, otherwise it is not a suitable cloning vector. Alright, so this comes to an end, and we get to know about one more tool that is useful in recombinant DNA technology.